Welcome back. In this video, we are going to continue our discussion of VDJ recombination, focusing now on molecular mechanisms, the sort of molecular genetics of recombination, and introduce ourselves to many of the key enzymes and other factors that accomplish the process of putting together an antigen receptor. And again, we'll focus in this video on immunoglobulin recombination, but as we'll see, most of the principles we cover today will be equally true for T-cell receptor recombination as well. So let's remember that in order to put together an antigen receptor, as we saw in the last video, that is going to involve the recombination of gene segments in the germline DNA. And so in this example, let's imagine we're putting together the light chain. So we're either in the lambda locus or the kappa locus. We have V and J segments. Once the cell sort of randomly, stochastically chooses one of those V's and one of those J's, in order to recombine them, to actually put them physically together, a few things need to happen. One is that we sort of need to bring them physically together in space because in the meantime, we have all these intervening sequences that are separating them. So the first enzyme that we'll learn about today is an enzyme called RAG, whose name comes from the name recombination activating gene. Of course, the gene is what encodes RAG. RAG itself is a protein and is a recombinase protein because it facilitates the process of recombination. So one thing that RAG does is it actually binds to the gene segments that we want to use for recombination and brings them physically together with on the DNA strand. And so we call this process synapsis from the idea of a synapse where you have two things sort of in close physical proximity to each other. And notice that when we synapse the two segments, the 1V and the 1J, that all of the intervening DNA is sort of looped out in space out here. And in fact, one of the other things that RAG is going to do is cut off that intervening DNA. And when it is cut off, it is left in a piece of DNA that we refer to as the signal joint, or at least this piece of DNA is sort of fused back together by something we call the signal joint. And what's remaining we call the coding joint. A key idea here is that the coding joint is the piece of DNA that is actually going to code for, in this case, the light chain, or in general, it's going to code for whatever chain of the antigen receptor in question it is that we're trying to make. Whereas the signal joint is going to be non-coding. That probably makes sense. As you can see here, it's a circle of DNA that's not very much good for transcribing RNA off of. And it gets its name as being the signal joint because it contains the original signals that told RAG where to bind and where to initiate recombination. And so it just, starting from a bird's eye view, this is essentially what we're about to see is how this process of bringing two segments together, synapsing them, cutting off the intervening DNA, leaving us with a piece of DNA that will then encode whichever chain that we're trying to create. All the steps involved, all the molecular genetics involved through this process. But this, again, is just a bird's eye view. So let's start by looking in more detail at how this occurs. So a key piece of information to know is that for any V, D, or J segment to recombine, it needs to bind to the enzyme RAG. And how does RAG know where the V, D, and J segments are in the genome? How does it know where to bind and what to bring together with what? We also don't want sort of random recombination of the incorrect segments to one another. So RAG relies on sequences in the DNA known as recombination signal sequences, or RSS. And as you can see here, all V, D, and J segments have at least one recombination signal sequence. In the lambda light chain locus, what that looks like is on the sort of downstream side of the V segments, they have an RSS, which you can see here has the number 23 in it. Whereas the J segments in the lambda locus, on their upstream side, they have an RSS that has this number 12. These numbers indicate something about the size of the recombination signal sequence. So if you look here in this box, you can see the structure of the recombination signal sequence. And all recombination signal sequences are made up of something called a heptamer and a nonamer. This refers to it being 7 bases long and versus 9 bases long. And what's key here is that the heptamer and the nonamer are conserved sequences, meaning that every recombination signal sequence has this exact heptamer, CAC, AG, TG, and the complementary sequence, and so on for the nonomer, this exact sequence on both sides. That's important. You need a conserved sequence because that is the part that RAG actually recognizes so that it can bind. That's how it knows that it's at the end of a V segment in this example, and that knows where it is going to bind there. The other piece of the recombination signal sequence, though, is called the spacer. And the spacer is made up of a non-conserved sequence of nucleotides. And in this example, we have a 23 base pair spacer, whereas other recombination signal sequences, again, they have a conserved nonomer and heptamer, but they have 12 base pair spacers. And we call them spacer because the point of this region of the recombination signal sequence is actually spatial in nature. What has been discovered is that 12 base pairs basically constitutes one turn of the DNA double helix, whereas 23 base pairs constitutes 2 turns of the double helix. And that's helpful because, we always draw these diagrams to kind of show the DNA in 2 dimensions. It's being stretched out on a line. But recall that DNA is a double helix. It has a 3-dimensional structure. And so for RAG to be able to bind a V segment and then a J segment and sort of hold them in the right physical orientation to each other to recombine them, it sort of needs to bind to one side of the helix and then the other side of the helix. And so these spacers sort of facilitate that 3-dimensional conformation of the RAG enzyme being bound to the DNA. In any case, however, what's another key portion or key idea here that you need to know is that RAG follows something called the 23-12 rule, meaning that RAG will only recombine segments if one of them has a 23 and the other has a 12. You never recombine segments that have the same spacer length in their recombination signal sequence. Now, fortunately, in these loci, you'll see that that's not hard to accomplish. So in the lambda chain locus, the V segments have 23 base pair spacers and the J segments have 12 base pair spacers. So we can recombine them using the 23-12 rule. In the kappa locus, that association is reversed. The V segments have the 12 base pair spacers and the J segments have the 23 base pair spacers, but that means they're compatible with each other to be recombined. Whereas in the heavy chain, recall that we have VD and J segments. You have an additional segment here. Notice that the D segments have recombination signal sequences on both sides. That's because they need to be recombined in both directions. The D segment needs to join with the J segment, and then that combined DJ segment is going to use this recombination signal sequence on the upstream side of the D to be recombined with V so that we get a final VDJ combination. So the 23-12 rule is important. It governs which segments can be recombined with which, and it also serves a sort of a conformational, a role in making sure that the shapes line up in a way that's going to facilitate recombination. But assuming that RAG has bound and it's found the sequences that it's looking for in terms of the recombination signal sequences, what happens after that? Well, this enzyme RAG, which is made up of 2 subunits, RAG1, 2, remember that it is the recombinase that's going to facilitate recombination. What you can see in this cartoon is that it has basically 2 active sites, 2 openings here, each of which will bind to the recombination signal sequence of an individual VD or J segment. So here it looks like we're recombining a light chain. So we're going to put a V together with a J. You can see that the recombination signal sequences, one is purple, representing a 23 base pair spacer. The other is orange, representing the 12. One half of RAG is bound to the 23. The other half is bound to 12. So it's obeying the 23-12 rule. And when it binds to those 2 segments, you can see that it brings them together in space. It is accomplishing synapsis. It is synapsing those 2 segments. And what this cartoon is representing is that all of the intervening DNA is being looped out. And this kind of double slash line is representing a discontinuity in the DNA, meaning that there could be a huge a huge amount of DNA that's being represented but not pictured in the image. Because, if you pick the very first J segment and the very last the very first V segment and the very last J segment, you might need to cut out quite a lot of DNA in order to recombine those two very physically distant segments with each other. But however much DNA is being looped out in this example, we need to get rid of it. And so the other thing that RAG is going to do is cleave the V and the J segments at their recombination signal sequences. So notice these triangles are sort of being cut off. So most of the recombination signal sequence then will actually be removed from the V and the J segments. And a couple of things are going to result from this. One is that we have now formed a coding joint and a signal joint. The coding joint is the piece of the DNA which constitutes most of the genome that maintains the V and the J segment. They are what are going to encode for the protein that we're trying to create. And at the coding joint, this cut DNA is then going to be stabilized by some DNA chaperones called Q70 and Q80. So we need them just to kind of like hold things together just for a minute before we can join the DNA back together, because we're going to need to repair it where it's been cut. In contrast, that other piece of DNA that I refer to as the signal joint, it gets its name from the fact that the recombination signal sequences primarily remain in that piece of DNA. And similarly, the signal joint also will be transiently stabilized by Q70 and Q80, so that we can process this DNA for later sort of recycling. But another thing to draw your attention to here is that the cut ends up generating two different types of endings on both the coding side and the signal side. And what's most important for our purposes is to notice that the cutting off of the recombination signal sequences results in two hairpin structures on both sides of the coding DNA. And those hairpins are covalently closed. The reason that matters is because that is going to be a challenge for putting the DNA back together. You can't just sort of mush two hairpins together. They're covalently closed off from one another. So we're going to actually have to reopen those hairpins in order to put the two DNA strands back together efficiently. And so we'll come back to these covalently closed hairpins in just a second. Whereas on the signal side, we do not form hairpins, and you have a more open, or what we call blunt ends to those pieces of DNA. So moving forward from this, we'll start by just talking about what happens with the signal joint, because it's sort of less important. At the signal joint, we have these open blunt ends that are temporarily stabilized by Q70 and Q80. And then we use a combination of DNA repair enzymes, including DNA ligase, which from its name, you can tell it's going to ligate the DNA, kind of tie it back together. That's what ligation is about. With its partner XRCC4, another DNA repair enzyme. And that's going to give us a kind of circular loop of DNA that we refer to as the precise signal joint. And this term precise will become a little bit relevant later, but what it means is that it's unaltered. It is precisely the DNA that we started with, meaning that the recombination signal sequences have not been altered. They've just been cut off, and now they are part of this piece of DNA that we refer to as the signal joint, which ultimately does not code for anything and will go on to be destroyed and recycled, and we'll reuse those nucleotides for other things later on that are irrelevant to the recombination process. So that's what we do with the signal joint. But now let's return and think about the coding joint, which again is what we're focused on. In the coding joint, as I said, Q70 and Q80 is going to stabilize those two open sort of ends of the genomic DNA that we want to keep. But we have the problem of there being these covalently closed hairpins. So the first thing we need to do is to open those hairpins. And the enzymes that facilitate the opening of the hairpins are DNA PK and Artemis. And so these are enzymes that can actually open up the phosphodiester bonds, the covalent links that form those hairpins. And now that the hairpins have been opened, if you sort of follow this diagram now back up to the top on the right side, now we can begin to facilitate the process of DNA repair. And so multiple enzymes come in to accomplish that. The first of those is terminal deoxynucleotidyltransferase, or TDT. TDT, it's going to start to transfer new nucleotides. And as we're going to see, it's going to actually add new nucleotides between the two segments that we're recombining. In this example, it was a V and a J. So it's going to add a few new nucleotides, which are represented in this image by this kind of little rainbow of colors. This represents new genetic information that's been added by TDT. Then additional enzymes, again, DNA ligase and XRCC4, are going to do some additional repair processes that sort of repair the backbone and put these two strands back together and formally kind of ligate them into a functional unified piece of DNA. And what we are left with then is called the imprecise coding joint, because it is not precisely the genetic material we started with. It includes additional random nucleotides in between. And this is where the idea of imprecision comes from. We have our original V and J segment with a few extra nucleotides. And as we're going to see, that's going to add some additional genetic information and result in the addition of one or more new amino acids, the addition of one or more new codons into the sequence. And so, as you might imagine, that is going to be a source of additional diversity in the possible final outcomes of the proteins that might be made. So what we're going to do now, though, is look a little more carefully at some of these processes, the activity of some of these enzymes, and how we actually, at a more zoomed-in level, put the strands back together once we reopen them. So here we are looking at the process of synapsis, where the recombination signal sequences have been brought together. The process of synapsis is going to involve cleavage of the recombination signal sequences, recall, and most of the recombination signal sequence ends up in the signal joint. But a small piece of it is going to remain behind, and that is going to result in some random nucleotides being added to the ends of the D, or in this example of the D and the J segment, but the same would be true with the V segments as well. And you can notice here these covalently closed hairpins. Well, we have to open them, and you might imagine that we would open them by just sort of cutting here, right at the end. You might just imagine it looks very sort of tempting that maybe you could just take scissors and snip that open. But that is not actually where DNA PK and Artemis cut. They, in fact, cut actually proximal to the segment, in this case, the D segment. So you can see that the cut happens here and here, proximal to the segment. And so what that means is that the the nucleotides on the that are next to the cut actually flip up and become part of the opposite strand. And so again, if you follow this diagram up here, you can see that now this A and G now become part of the strand above, and here this A and T flip down to become part of the strand below. These new nucleotides we refer to as P nucleotides because we've now formed palindromic sequences. Because originally these two nucleotides were complementary to the two nucleotides above. Now that they are found kind of on the other side, that creates a sort of synonymous sequence if you read it front to back, and that's what a palindrome is. Even if you don't really get the idea of a palindrome, that's okay. It's most important to know that the opening of the DNA hairpin just generates some new random nucleotides on both sides, both strands, and we refer to those as P nucleotides. So Artemis and DNA PK, their activity yields P nucleotides that have been now added to the sequence. And so that will be a source of some additional genetic information, as I said. However, we add even more nucleotides after that through the activity of TDT, terminal deoxynucleotidyltransferase. It is now on the ends of those P nucleotides going to add even more random nucleotides. And the random nucleotides that are added by TDT we refer to as N nucleotides. So you can see that both strands now have a good number of additional nucleotides that have been added. And TDT adds kind of a random number of nucleotides. And depending on the chain that we're recombining, that number might be very low or it might be very high. Anywhere between 0 and around 20 nucleotides get added by TDT, depending on context. So this is cool. We're closer to where we wanted to be. But notice now that we have one strand of DNA that kind of has this overhang of a bunch of nucleotides. and then the other piece of DNA kind of has, maybe we could think of it as an underhang. But DNA needs to be double-stranded. So we need to be able to like put these two pieces of DNA back together to re-anneal them so that the DNA is back in the kind of double-stranded conformation that it needs to be in. And it would be nice, you could sort of imagine that, uh, it looks kind of like you should be able to just kind of like, okay, just take the overhang and take the underhang and just mush them on top of each other. But unfortunately, because these nucleotides that we've added are random, it's very unlikely that this whole sequence is going to be complementary to this whole sequence on the other side. So what happens instead is that we sort of take the strands and you kind of just like rub them against each other. They just sort of randomly are moving, like molecules do. There's some Brownian motion involved. And they sort of test themselves against each other through just kind of physical association until sort of by chance you get 2 or 3 nucleotides that are complementary to each other within both sequences. And that is usually enough to anneal the 2 strands together and anchor them together long enough for the processes of DNA repair to kind of kick in. So in this example, we started with sort of TCA and TAG, but this TC sequence and this AG sequence, they are complementary. And so the DNA will now anneal at those 2 positions. But notice we have a couple of problems. One is that this A is now not complementary to this C, nor is this T complementary to this C. So we need to get rid of these mismatches. So we will use exonuclease enzymes to perform mismatch repair at these positions. But the other problem now is that even with these mismatches removed, we still have open parts. We have gaps in the DNA on both strands. And so additional DNA repair enzymes will then fill in the missing holes that are generated by this process, yielding a complete sort of double-stranded set of DNA. And what we are left behind with then are these palindromic nucleotides or P nucleotides that were generated by DNA, PK, and Artemis, as well as some number of N nucleotides whose origin was from the activity of the enzyme TDT. And again, the exact numbers of enzyme or of nucleotides that you end up with varies from recombination event to recombination event. But one way or the other, we've now introduced some number of additional nucleotides between the germline segments that we recombine. So in this case, between the germline D segment and the germline J segment, this is completely new genetic information that has been added here on the spot, kind of de novo. And it is going to constitute new codons, which can then encode for new amino acids. And those amino acids then are going to change the way that the final immunoglobulin chain is going to fold and therefore change its final shape, its final structure. And of course, as I've said many times in these videos, it is the shape that is going to confer the function. It's the shape that's going to determine which antigen that immunoglobulin can bind to, and therefore its part in the overall immunoglobulin repertoire. And so a term that we should introduce ourselves to now is that the additional nucleotides that are added by P nucleotides and N nucleotides, we refer to this as junctional diversity. And so we add the junctional diversity generated through recombination on top of the combinatorial diversity, and that is what gives us the overall diversity of the entire immunoglobulin repertoire. Another point to make, though, is that, as you can see, there's a lot of random things happening here. And anytime we rely on random processes, there's usually a lot of waste and a lot of error that can occur in that process. So one thing that I want to draw your attention to is that the recombination of V, D, and J segments is only good, or it's only a good outcome about 1/3 of the time. About 33% of recombination events are successful. That is because in order to generate a functional protein, you need to add a number of nucleotides that will introduce new codons without inducing deleterious mutations into the sequences downstream. So there are a couple of mutations that might be introduced by the random addition of new nucleotides between the germline segments. So one that's very obviously bad is that if you accidentally, through the addition of random nucleotides, introduce a stop codon, If you accidentally introduce a stop codon, that is going to terminate translation at all of the genetic material downstream of wherever that stop codon is introduced. So here you can see we're recombining D with V, and that the combination of P and N nucleotides randomly introduced a stop codon. Therefore, nothing is going to be translated downstream. This chain will never fold and will never be able to serve as a functional heavy chain, and that will represent a kind of failed recombination. And the B cell will have to try again, and if it never does successfully recombine a heavy chain, ultimately it will die because there will be no point in keeping it around. But the stop codon is not the only deleterious mutation that might result. The other mutation that might happen is what we call a frame shift mutation. And frame shift mutations occur when the numbers of P and N nucleotides add up to a number that is not a multiple of 3. Remember that codons are sets of 3 nucleotides that together encode a single amino acid. And so in this example, or in the good example, we added 12 nucleotides, which represented 4 codons. And those 4 codons introduced tyrosine, serine, glycine, and serine into this region between whatever was being encoded by the V and the D segment. And that's great. We've got 4 new amino acids. They're going to fold in whatever particular way, and that's going to introduce diversity. However, if we add a number that is not a multiple of 3, in this case like 13, we'll still get some amino acids, but we'll change the reading frame of everything downstream, in this example in the D segment, and then also all the way through into the constant region of the heavy chain as well. So frame shift mutations are bad news because that's going to change the way everything else downstream is going to fold. And usually that means that it is not going to fold in a way that is actually functional. So the overall implication of that is that we need to add multiples of 3 nucleotides to have a productive or successful recombination. So we get a lot of recombination events that fail and that are wasted, but we have a lot of B cells, as you saw, 10 to the 11th at any given time. So we have so many cells that we get a lot of shots at this, and it happens successfully enough that we still generate overall a very diverse repertoire of functional immunoglobulins expressed within our B cells. Okay, we can summarize this material. We saw that gene segments are recombined at the molecular level with the help of an enzyme known as RAG, and RAG knows where to bind through the recognition of things called recombination signal sequences, or RSSs. And RAG follows something we call the 2312 rule. It binds to gene segments, one of which needs to have a 23 base pair spacer, the other of which needs to have a 12 base pair spacer. And when it does that, it can synapse those 2 segments and cleave in between them, or cleave them off and cleave off the intervening DNA, which gives us the coding joint and the signal joint. We focus mostly on the coding joint because that is what actually codes for the antigen receptor that we're trying to make. And we saw the activity of several key enzymes that facilitated the repair of the coding joint. So we saw that DNA PK and Artemis, they open up the closed hairpins that are generated by RAG, resulting in P nucleotides or palindromic nucleotides. The enzyme TDT adds new random nucleotides to those open endings, and we refer to those as N nucleotides. And then finally, the combination of DNA ligase and XRCC4 ends up facilitating the final ligation or closure of the double-stranded DNA strand to completely finish the process of repair. And all of these processes ultimately add up to the idea of junctional diversity. We form an imprecise coding joint. It is imprecise because we've added new genetic material to it in the form of P and N nucleotides, and this could add some number of additional amino acids, which could confer additional diversity, or it might result in deleterious mutation, like the introduction of a stop codon or a frame shift mutation in about 2/3 of cases. So we rely on sort of the law of large numbers to overcome the fact that our success rate is relatively low, but nevertheless, it's high enough that we put together a really staggering number of unique antigen specificities within our immunoglobulin repertoire. So that covers it for part 2 of our discussion of VDJ recombination. In the next video, our final discussion of VDJ recombination, we'll look at the ways that these processes play out in T cells, including the ways that it's mostly similar in T cells, although there are some unique considerations that we'll cover there too. See you in the next video.